Cloning in Saccharomyces and other fungi. Although the process of cloning was started in 1970s with other organisms, especially bacteria, but for fungi, the process of cloning was started in late 1970s. And at that time, the preferred organism for cloning was yeast. So at that time, the main purpose of cloning with yeast was that to know the function of other foreign genes in vivo within the cell. And in addition to that, secondary purpose was that what functions of eukaryotes, especially like yeast, they are having with reference to some related genes like जो function थे related to differentiation of the cell and then signal transduction. इसके अलावा जो सेल डिवीजन के प्रोसेसेस हैं लाइक माइटोसिस या मियोसिस, तो इस तरह के जो फंक्शंस जो थे वो मेनली स्टडी किए जाते थे. बट विद इन ए फ्यू डेकेड्स देन द पर्पस फॉर क्लोनिंग विद फॉर यीस्ट सेल इट इज टोटली चेंज्ड. जो यीस्ट है इसका जो फर्स्ट टाइम फुल जीनोम सीक्वेंस किया गया था वो अराउंड 1996 में था, जब इसका कोई अराउंड 12 मेगाबेस जो जीनोम है इसको सीक्वेंस किया गया. तो इससे हमें बहुत सारी इंफॉर्मेशन मिली कि जो ज़्यादातर जीन्स थी हमें उनकी जो फंक्शंस हैं उससे हमें आगाही हुई कि व्हाट टाइप ऑफ फंक्शंस दे आर परफॉर्मिंग विद इन द सेल. In addition to that, the interaction of different proteins within the cell that are encoded by different genes that was also studied, and especially under what circumstances the genes can be expressed. So all these answers can be studied if we are going to perform gene manipulation in the yeast cell by using different techniques. Then the main purpose for cloning by using yeast cell was to produce some commercial proteins, that is overproduction of proteins or some industrial products by cloning specific genes within the yeast cell, because by using some specific strain of yeast we can overexpress certain products. Now those products are specially related to food industry or beverage industry, so that we can control the desirable properties of our food products or beverages. In addition to yeast, some other fungi like the Pichia pastoris, it is a type of methylotrophic fungi. Methylotrophic fungi are those fungi that use single carbon sources as nutrition, like the methanol or methane. So such fungi, they are also a good source for cloning foreign DNA. Another reason for cloning in yeast was that when cloning was attempted first time with bacterium by using different type of plasmids, then the length of the foreign DNA that can be encoded, that was also very important, because initially very small DNA fragments, they were cloned in bacterium. But in order to enhance the size of foreign DNA for cloning, some other organisms were required, and for that purpose yeast artificial chromosomes, they were developed that have the high ability to clone foreign DNA that may be up to more than 1 megabase. So because of all these reasons, gene cloning for yeast and other fungi, they are preferred.